Hello everyone and welcome to Chapter 4 Cell Membranes and Transport. This is the first video of 3 parts. Now in this chapter we'll be learning about, cell membranes and how it looks like and how it functions and transport across the cell membrane. So there are 3 parts in this chapter. Part 1, we will learn about the structure and function of all these different components. Part 2 will be about cell signaling. And this is a way cells communicate and cell send signals to other cells. So cell signaling, there's a process right there. And part 3 is transport across the membrane, where we will explore these 5 different types of ways things can be transported across the membrane. So let's start with part 1 now. Part 1 is all about cell membrane structure and function. Now, as we know from chapter 1, the cell surface membrane or plasma membrane is around 7 nanometers thick and is primarily made out of this phospholipid bilayer. And this is a phospholipid little diagram right here, and the heads are called hydrophilic heads, and they point outwards, and they have these tails which point inwards. These are hydrophobic tails. Let me draw a line there. Hydrophobic tails. And because they all point inwards, this forms a hydrophobic core. Now, what is the function of the cell surface membrane? In general, it is to control movements of substances into and out of the cell. And how it does this is because it is semi-permeable. The cell surface membrane is considered partially permeable to substances. It is a barrier to water-soluble substances, mainly because water-soluble substances cannot pass through the hydrophobic core. It cannot interact with the hydrophobic tails here in the hydrophobic core, so it cannot pass through the cell surface membrane. However, this cell surface membrane made of phospholipid bilayer allows passage of lipid-soluble substances. Why? Well, lipid-soluble substances can pass through the hydrophobic core and definitely interact with it. So these are the general kind of descriptions when we talk about cell surface membrane in general. However, we can also say that the cell surface membrane or the plasma membrane has many more roles, such as cell signaling, cell recognition, cell-to-cell adhesion. It could be site for enzymes to catalyze reactions, anchoring for the cytoskeleton, and even form hydrogen bonds with water for stability. And the reasons why it has so many of these other roles is because the membrane is not just composed of phospholipid bilayer. It is also made out of other components. Now, before we look at other components and its structure and function, we really need to talk about the fluid mosaic model. We say that the cell surface membrane follows the fluid mosaic model. And like many definitions in biology, we can kind of take the 2 words apart and define it individually. So we define fluid first, and then we define mosaic. This is how we should write your answers as well. Define fluid first, and then define mosaic. So what is fluid? Fluid refers to the phospholipids and protein molecules that are able to move about and diffuse sideways within its monolayer. Okay, this is a bilayer, and this here is a monolayer, right? So what is it doing? You can see the phospholipids here and proteins here, how they are moving sideways. There is some movement there. It is fluid. It is not like a rigid solid thing that doesn't move at all. Okay, so it is fluid. It can move sideways within its monolayer. Okay, what about mosaic? What does mosaic refer to? Mosaic refers to proteins interspersed or scattered within the membrane. And we can see the proteins represented here in this green blob and this white blob here. And they're moving around, they're scattered around the membrane. Now, in a membrane, there are many different proteins. And therefore, if you look at the cell surface membrane, right, using a scanning electron microscope, so you look at it, the 3D surface of the cell, it wouldn't be quite smooth. It will be quite bumpy, and this would actually give rise to a mosaic kind of pattern. These bumpy bits on the cell surface membranes are basically proteins interspersed or scattered within that membrane. So yeah, this gives rise to the term fluid mosaic model. Our cell surface membrane follows the fluid mosaic model. Okay. Now that we're done with this model, we are ready to look at the different components of the plasma membrane and the different functions of them. Number 1 is phospholipids, obviously. Number 2 is cholesterol, 3 proteins. Number 4, glycolipids. So glyco refers to carbohydrate. So this is carbohydrate plus carb plus lipid, and glycoproteins. Glyco refers to carbohydrate as well. This is carb in combination with proteins. So each of these components would have slightly different functions, and we will go through them one by one, starting with phospholipids. This is a little bit of a chapter 2 recap. All we need to know is, hey, look, this phospholipid is made up of a glycerol, 2 fatty acids, 1 phosphate group, and maybe, it could be modified at this R group right here. There might be other groups attached to it. Now, because of this structure, phosphate group is charged and fatty acid tails are definitely not charged, right? So because of this chemical structure, this results in a hydrophilic head and a hydrophobic tail. So what is the role of a hydrophilic head? Hydrophilic head has a phosphate group, is charged polar, and it's able to form hydrogen bonds with water, and this allows the phospholipid to stabilize the membrane. So the hydrophilic head, it forms hydrogen bonds with water and stabilizes the membrane overall. Now, hydrophobic tails, on the other hand, are made of fatty acids, and they are non-polar and hydrophobic, so it does repel water. Now, because of this hydrophilic head and hydrophobic tail, if you take phospholipids, so just phospholipids, just a bunch of them somehow, and you dump them into water, they're gonna give you a few arrangements, and the arrangements would probably look something like this. The phospholipids would self-assemble because of its nature. Hydrophilic head, hydrophobic tail, right? Because of its nature, hydrophobic tail is going to point away from the water. Hydrophilic head is going to point towards the water. So you could form a bilayer like this, or if there are too few of them to form a bilayer, it could form a micelle. The micelle here, you can see it's just one layer. Actually, it's not double layer like this. It's just one layer. And you can see how there is no water inside, because this, in the middle of the micelle, is where all the hydrophobic tails kind of direct to, so repel water, point towards each other, whereas the hydrophilic head interacts with water. Micelles, yeah. So inside, sometimes in the medical world, they use this micelle structure to carry drugs inside of it and deliver it to patients. Some, not all. You can Google and learn more about that. Anyways, what is the point here? The point here is that phospholipids have a hydrophilic head and hydrophobic tail, and this allows it to self-assemble into different structures when you dump them to water. Okay, but what is the role of the hydrophobic tails? Hydrophilic head, oh my gosh, it's so easy to mess up. Hydrophilic head forms hydrogen bonds with water and stabilizes the membrane. How about hydrophobic tails? So hydrophobic tails, since it can form the hydrophobic core that we have talked about just now, this helps the phospholipid bilayer to be a barrier towards water-soluble substances and allow passage to lipid-soluble substances. So if the molecule is lipid-soluble, small, and uncharged, then only it can diffuse through the phospholipid bilayer. Now, there's also an extra function for the tails. The fatty acids can also help maintain fluidity of the membrane. What do we mean by fatty acid tails can maintain membrane fluidity? What even affects membrane fluidity? So here are a few things that can affect membrane fluidity. Number one is temperature. So think about it. Membrane fluidity is about how the phospholipid kind of moves around sideways, right? So if temperature is high, kinetic energy is higher, and therefore those phospholipids will move around more quickly, randomly. So that temperature would affect membrane fluidity. Number two, ratio of unsaturated to saturated fatty acids. Now, the more unsaturated fatty acids there are, as we have learned in chapter 2, unsaturated fatty acids have a carbon-carbon double bond, at least one or more. Okay, and if there are more carbon double bonds, this causes kinks, bends in the fatty acid tails. And therefore, if you have more unsaturated fatty acids, then the ratio of unsaturated to saturated fatty acids will increase. And therefore, because of the kinks, there is more space. There is more space between individual phospholipids and cause them to be more loosely arranged. And if there's more space between phospholipids, it's going to make the membrane move around even more. So it's going to become more fluid. However, the reverse is true. So if there's more saturated fatty acids, this would lower the unsaturated to saturated fatty acids ratio and make the membrane less fluid. Why? Because saturated fatty acid does not have this C double bond and does not have kinks, and therefore there's more area for phospholipids to interact, more area between those phospholipid tails to interact, and therefore they can pack together even tighter and lower membrane fluidity. Now there's one more thing that can affect membrane fluidity. Phospholipid tail length. The longer the tails, the less fluid it is. Why? Because it's all about surface area. The more surface area, then the more interaction these tails would have with each other, and this allows it to be more to be less fluid. Longer tails, less fluid. Last but not least, what affects membrane fluidity is also cholesterol. So cholesterol can also maintain membrane fluidity both at high temperatures and low temperatures. And we'll see that later. But what you should take note is this. What is the point of regulating membrane fluidity? Now, your cells will be exposed to different temperatures, right? Sometimes higher or lower temperature. The cell has this optimum fluidity that they have to be at, a very sweet spot of fluidity that they have to be at in order to function optimally. So, if they are exposed to different temperatures, okay, so like higher temperature, it will make the membrane more fluid, right? So that's not ideal for the cell. However, the cell can maintain fluidity by changing ratio of unsaturated to saturated fatty acids, the length of tails, and adding cholesterol. Okay, so again, if we have high temperature, it will be more fluid, correct? So the cell would want to strive to lower the fluidity because it's too fluid now. It's not optimal for the cell to function. So at higher temperature, they will most likely increase the number of saturated fatty acids in the membrane to make it less fluid. Okay, and the cells may also make the tails longer, so it's less fluid at higher temperatures, and maybe add some cholesterol. All these to counteract, to make sure that it's still functional, that the fluidity is maintained at an optimal state at even a higher temperature. Okay, that's just an example. So let's look at cholesterol now. How does cholesterol actually regulate membrane fluidity? Now let's look at the structure first, okay? Cholesterol is actually quite a small molecule, and you can see how it compares to the other phospholipids here. It has a small hydrophilic head, which is like just this OH group right here, and also has a hydrophobic tails. And this allows it to fit between the phospholipid molecules. And you can see it here, how it fits between them. The hydrophilic head and of the phospholipid is interacting with the hydrophilic head of the cholesterol, and the tails are interacting with each other. The tail is made out of this 4 rings, 4 steroid ring structure, which you do not need to know in detail. Just need to know it looks something like that. Okay. Cholesterol is not found in prokaryotic membranes, only in eukaryotes. So think of humans mostly. Okay. Now that we know how it looks like, how it fits into the membrane, what roles does it play? Now, as we said just now, it helps regulate fluidity of the membrane, and actually overall it stabilizes the membrane. And because it's right in between different phospholipids, this can cause it to have lesser space and block passage of very small ions through the membrane. Okay, let's explore regulation of fluidity. How does it do that? Now, let's have 2 scenarios. At lower temperatures, the membrane is less fluid, correct? Less kinetic energy, less fluid. So how does cholesterol function? It increases fluidity. So it pushes it back up to an optimal kind of fluidity. And it does this by preventing close packing of the phospholipid tails. So because it's hanging out in between these phospholipid tails, it gives them a bit of space so the phospholipids can move around. It's all about the packing, yeah? How about if there is high temperatures? So high temperatures, higher kinetic energy, more fluid. So it's too fluid, you want to decrease the fluidity. So cholesterol does that, but how? Cholesterol decreases fluidity by reducing the mobility of phospholipids. So it kind of, because it's like between those phospholipids, so the phospholipids kind of have a restricted space to move. And therefore, this could decrease fluidity. So cholesterol basically has different functions, different regulation functions during high temperatures and low temperatures. Slightly different explanation, but overall, it stabilizes the membrane, it maintains, regulates membrane fluidity, whether the temperature is high or low. Don't forget though, like we learned just now, that cholesterol is not the only factor that can stabilize the membrane. The cell also can, change the length or the saturation of fatty acids in order to regulate membrane fluidity. So yeah, roles of cholesterol: regulate fluidity, very important point, stabilizes the membrane, very related to regulating fluidity, and number 3, block passage of very small ions, and that's because it's, kind of crammed in between 2 phospholipid molecules here, and therefore small ions may not be able to pass through because there's less space now. And that's how cholesterol functions. We're done with phospholipids, we're done with cholesterol now, and the idea of membrane fluidity. Now we are ready to talk about membrane proteins. So, membrane proteins. There are types based on their position in the membrane, and we have different names based on their position. If they're hanging out on the inner or outer surface of the membrane, we call them extrinsic or peripheral proteins. So they're hanging out at the side, they're just at the surface. And we can see them here, extrinsic, so at the side. This here is also an extrinsic protein. Now, how about those proteins that are embedded into this membrane. So this here, even it's halfway through or all the way through, we call them intrinsic or integral proteins. It extends into the hydrophobic core of the phospholipid bilayer. Now, these proteins may be mobile or fixed. So yeah, some proteins can move around laterally, like in the fluid mosaic model, but some are actually fixed structures, okay? And we have a special name for those who kind of extend like all the way from one side of the membrane to another. So we call that spanning across the membrane. The special name here is transmembrane protein. So this golden color protein here is an intrinsic protein, which is a transmembrane protein. So yeah, one example of transmembrane protein you may know is transport proteins. All transport proteins are transmembrane proteins. Now, for all intrinsic proteins, as we said just now, it extends not only on the surface, but extends into the hydrophobic core. So, in order for it to stably be held there, they would have to have both hydrophobic and hydrophilic regions. These are structures of all intrinsic proteins, no matter their function. So whether if they are like transmembrane or not transmembrane, or whether maybe they are transport protein or an enzyme, it doesn't matter. As long as they are intrinsic protein, they would have a structure that looks like this. They would have both hydrophobic and hydrophilic regions to be stably placed at the membrane. The protein would have hydrophobic regions that interacts with the hydrophobic core or the fatty acid tails of the phospholipids. Here you can see in lighter purple here, and you can see how this hydrophobic region can interact with the hydrophobic core. Now, how about the hydrophilic regions? And that's kind of common sense by now, right? So hydrophilic regions would hang out, interact with the hydrophilic heads and able to extend into the aqueous external environment inside or outside the cell and interact with water. Now, intrinsic proteins all have this structure, no matter the function. And this again allows it to be stably held the membrane. It won't like repel water suddenly and like slip out of the membrane, right? It's gonna be stable because of this kind of regions in the protein. So yeah, this is the structure of intrinsic proteins. Now let's talk about roles. Those are the structure roles. Now, one of the most famous roles we all love is transport proteins, and you guys should be familiar with this term already. Now, because the phospholipid bilayer is partially permeable, you do need transport proteins to allow passage of ions, charged polar or larger molecules through the membrane. These cannot pass through the phospholipid bilayer, so they require transport proteins. There are generally two types of transport proteins: channel proteins or pore proteins, and usually this is used for facilitated diffusion. We'll learn more about facilitated diffusion later. And carrier proteins, which are involved in both facilitated diffusion and active transport. Let's look at the structure of channel proteins. Channel proteins are highly specific, so if its structure is meant for that particular ion, they will only transport that particular ion. For example, I don't know, hydrogen ions. So it will only transport hydrogen ions. So it's very specific for that particular substance. And yeah, this channel or pore of the channel proteins are often water-filled. No, it's always water-filled. And you can see that this pore extends from, one side of the membrane to another. It's just like a hole, right? And it's water-filled. And this is kind of enabled by hydrophilic R groups on amino acids that face inwards towards the channel. Now, as we said just now, all transport proteins are transmembrane proteins and are intrinsic proteins. So you would expect them to have hydrophobic regions and hydrophilic regions. Now, hydrophobic regions of the channel protein are obviously near the hydrophobic tails, interacting with the hydrophobic tails. Hydrophilic regions of the channel protein would be the outside area and the inside area, like interacting with the aqueous environment as well as the hydrophilic heads. Now, one more thing about channel proteins is that the middle, the pore, is also made of hydrophilic regions, which are basically R groups of protein. So hydrophilic R groups point inward towards the channel, enabling water to fill it and enabling water-soluble substances to pass through. Now, an example of a channel protein, which you will hear about a lot in your syllabus, are aquaporins. Aquaporins are channel proteins for water. Now, water is one of those exceptions of polar molecules that can pass through the phospholipid bilayer. But for water to pass through the phospholipid bilayer straight is kind of tough, right? Because it is dipole in nature, and you know the hydrophobic core is hydrophobic. However, water is small enough to squeeze through, and it's possible again, but it's a bit difficult. So for water to be transported really quickly, some cells have aquaporins, which are channel proteins for water, that allows water to pass through much quicker than through the phospholipid bilayer. So yeah, fun fact for you. You will hear about aquaporins more later on. Okay, more on structure of channel proteins. They can also be gated, which means they can open or close. Now, for example, a channel protein could be voltage gated. This means it can only open under a certain charge. So for example, maybe only if the membrane is negatively charged will it open. This happens a lot in neurons. How about ligand gated? So ligand-gated channel proteins would only open if a certain ligand or substance is present. For example, that ligand could be sodium ions. So that sodium ion has to bind to the channel in order for it to open to transport maybe something else. Without sodium ions, it's going to be closed and the channel protein will not be able to be put to use. Okay, so it can open and close. Sometimes. It kind of depends on the type of channel proteins. Yes. Now, how about carrier proteins? Now, one example we learned about carrier proteins is the sodium-potassium pump. And just like channel proteins, they are also highly specific. So this pump here, it can only transport sodium and potassium and not anything else. So sodium and potassium ions only. And we can see how it has a conformational change that occurs when it interacts with the ion or molecule. You can see here how the 2 potassium binds and then it changes shape. Okay, it binds, then the whole thing changes shape, and then it kind of, releases it to the side. So we say that the binding sites alternately open to one side of the membrane and then another. So it's not like channel protein where there's like a straight hole that goes right through it, right? It kind of flips between 2 conformations. It opens on one side, then it opens on the other. And they will constantly flip between 2 shapes. Now, as I said just now, we are looking at the sodium-potassium pump here, and this pump here specifically pumps 3 sodium ions out and 2K into the cell. So I say this really quickly in my head, 2K in, 3 na out, 3 na, 2 in, 2K in, 3 na out, right? So 2K potassium ions coming in, 3 sodium ions coming out. This is standard across all sodium potassium pump. Very useful to remember. There will be definitely questions on this in the past year. Okay, this direction is always the same in all cells. Okay, so that is channel and carrier proteins, and both of them are transport proteins at the membrane. However, not all membrane proteins are transport proteins. There is transport proteins, but there are also certain membrane proteins that are just enzymes. there are membrane proteins that are receptors for signaling molecules. So you can see the signal molecule here, and this binds to the receptor, which then results in some kind of process that happens. Number 4, it could be anchoring the cytoskeleton. Now, if you remember from chapter 1, cytoskeleton is made out of microtubules, and some proteins at the membrane kind of like clips it in place, so anchoring cytoskeleton, and this would help maintain cell shape. Now, other than that, there are also proteins that are involved in cell-to-cell adhesion. So, you know, why is your organs not falling apart? That's because your cells do stick to one another in some way. Adhesion literally means sticking. So how do cells stick to each other? Some proteins do that. And you can see here, this is one cell, this belongs to one cell, this belongs to one cell. And you can see how proteins link to each other to allow the cells to stick together. Okay. So yeah, these are the roles for proteins, not just transport proteins, okay, but also all of these. Yes, you need to remember them. Yep, moving on. Now, how about glycolipids and glycoproteins? Now, glycolipids, as we have seen just now, did we see it just now? You have seen previously. Glyco refers to carbohydrates, therefore glycolipids really refer to a kind of a mixture between carbohydrate chains and lipids, sometimes phospholipids. Okay, phosphate group may or may not be there, but you can see here how the glycolipid like stays in the membrane, right? Because there is a phospholipid at the end, so phospholipid's there. The glyco part, the carbohydrate, kind of sticks out of the cell. Glycoprotein, on the other hand, is carbohydrate chains attached to a protein. You can see the protein here. This looks like an integral protein. It's a transmembrane protein, and this protein would have hydrophobic and hydrophilic regions as well. This area is going to be hydrophobic, and the areas facing outside is going to be hydrophilic. And you can see this carbohydrate chain kind of attaching to a protein, also sticking out of the cell. Carbohydrate chains all face the outside of the cell. And because they're not just some of them, there's actually quite a lot of them, this forms actually a sugary coat on the cell called glycocalyx. So yeah, that's how glycolipids look like. You will need to know how to recognize them in diagrams. Not very hard. Lipid, bottom, carbohydrate head, glycolipid. Now, what do they do exactly though? Okay, so because these carbohydrate chains kind of stick out of the cell, right? These carbohydrate chains actually interact with water to stabilize membrane structure. It's able to form hydrogen bonds with water molecules. Some parts of the carbohydrates, that is. Now, number 2, this could be for cell-to-cell adhesion as well. So glycolipids can participate in that. It could be cell recognition. Now, what is cell recognition exactly? So cell recognition is the idea that I can distinguish from my cells and foreign cells. So we say self and non-self. So what is my cells, my human body cells, and what is maybe a bacteria cell? How does the body does this? So the body does this by having certain glycolipids that act as cell surface antigens or markers. Now, antigens we usually we use this term to refer to foreign things like bacteria, viruses, pathogens, right? These other things have antigens, and we say that our body has markers. So our own cells have these cell surface markers. So slightly different terms, same idea, all made of glycolipid, okay, for cell recognition. Now, what happens when the body detects an antigen? Okay, what's an antigen? Again, it's a macromolecule in the cell surface membrane, in this case, a glycolipid, right? The carbohydrate chains look different on a bacterial virus. So for example, if someone injects you with a foreign bacteria, maybe just for example, right? Injects you with a bacteria, your body is going to look at it, okay, and there are receptors that read it and go like, okay, the carbohydrate chains for their glycolipids, okay, are not quite like our human ones. And therefore, this is considered a non-self. This is a receptor reading the antigen here, okay, or maybe the marker here. So it reads it and goes like, oh, okay, this is not normal. This is not myself. This is non-self, and therefore it would trigger an immune response to occur. So cell recognition is incredibly important. It is, again, to distinguish self from non-self. The carbohydrate chains of glycolipid, and later on you will see glycoprotein as well, they look different for your cells and others foreign cells. And therefore, your body knows when to trigger the immune response. Now let's look at glycoprotein. Glycolipid has these 3 roles, right? Glycoprotein has these 3 roles as well. They're the same, exactly the same, plus 1. So for glycoprotein, it has extra 1 function that is not for glycolipids. It is to be a receptor for cell signaling molecules. Proteins, not glycoproteins, some proteins do have this as well. They don't have carbohydrate chains, but they also have this function. Now, what do? Well, they kind of act as a receptor that receives signals from inside. This is the signal molecule. And that results in some kind of reaction. We will learn about this signaling pathway in the next video. But for now, I just want to draw your attention to this crazy diagram right here. I just want to remind you that for glycoproteins and proteins, okay, even though the carbohydrate chains are not shown here, right, for glycoproteins and proteins, and all proteins really, they are all made out of polypeptide chains. Now this here looks like we usually draw like an oval shape or blob that symbolizes protein. But I want you to understand that the protein is always made of polypeptide chains, which has a primary structure at first, a specific sequence of amino acids, and then it coils and folds into a 3D shape. And this one here is a tertiary structure. And you can see here how there's one polypeptide chain. So this is the beginning and the end, right? Beginning and end of the polypeptide chain, and it's like curving and folding throughout the membrane. These cylinders here actually represent barrels where the polypeptides are kind of coiled like this. So it's just a representative diagram. Instead of drawing the coils, they draw it as barrels. Okay, and we can see here how it's colored differently. This yellow part and this orange part, and there's a gray part. And that's because these different colored regions have slightly different properties. This gray region right here, it has hydrophobic regions. Why? Okay, the amino acids in these gray regions would have hydrophobic R groups, which can interact with the hydrophobic tails of the phospholipid bilayer. So that makes sense. However, this yellow region and this orange regions right here, these have amino acids which have R groups that are hydrophilic, which then can interact with the environment, aqueous environment, and the water, and the hydrophilic heads. So yeah, I just wanted to show you this diagram because it came out once before, but even though it looks incredibly complicated, the principle is still the same. Glycoproteins, proteins, they're made of polypeptide chains. Amino acids have different side chains, have different properties that interact with different parts of the phospholipid bilayer. So yeah, I hope that's kind of eye-opening. I hope you've learned something today. We have learned 5 components already: its structure and its function. So you should be able to pause this video and identify each and every component that is drawn here. With that, we've come to the end of our video. See you next video. Bye-bye.